Hello, my name is Gwen Soa. I'm professor and chair at the University of Pittsburgh, Department of Physical Medicine and Rehabilitation, and I'll be sharing with you some perspectives today about how we can use biology to inform precision medicine approaches for musculoskeletal care and in hopes of moving the needle in terms of improving outcomes for this challenging and large patient population that we serve. Disclosures are shown here on this slide. These relate to unpaid board of director positions, as well as our research funding, predominantly from NIH, PCORI, and some foundation and society grants. So really the key to success as we think about treatment for musculoskeletal conditions is the right treatment for the right patient at the right time. We've had a number of advances and innovations in the care of the musculoskeletal patient. However, what we've seen is that largely our outcomes have not significantly changed. And I believe that this is largely related to some lack of specificity in our treatment approaches and not matching appropriately the treatment for that particular patient to what's going on with their individual biology as well as their level of function. And in order for us to be able to address this, we're going to have to change our approach and really start to phenotype patients. And so what I'll share with you today are some examples of how we've looked at this in a number of different musculoskeletal conditions to try to better phenotype these patients and better rationally design their care to perfectly fit their exact phenotype. So the first question, as we think about the musculoskeletal system, is really what are we treating? So the entire musculoskeletal system undergoes wear and tear changes that accumulate over time and result in degeneration. And this degeneration results in pain and loss of function in this patient population. But what also happens over the same period of time are changes to the tissue structure and function related to natural aging. And while these changes related to wear and tear changes and age-related changes can look similar histologically or radiographically, what we have seen is biologically, they're quite distinct. And this is an example where we studied the intervertebral disc of the spine, and we compared what happened in a model of injury-induced degeneration to one of natural aging. And what you can see is there are significant differences in anabolic molecule gene expression, such as proteoglycans, growth factors, such as BMP2 and TGF-beta, and inhibitors of matrix breakdown, such as TIMP1. And so while these profiles anatomically might look the same, biologically there are very different processes at play. And so we clearly can't treat these distinct processes with the same treatment and expect to get a beneficial outcome. Similarly, across the entire spine, there are age-related changes that we see in this patient population. We see changes in the disc, we see changes in the bone, we see changes to the elasticity of the ligaments. We see arthritis of the small facet joints and hypertrophy. We see changes to the muscle and fatty infiltration in the muscle that accumulates over time. But this doesn't really tell the whole story. Just looking at what the musculoskeletal tissue looks like doesn't explain to us how the patient is functioning and how their patient is experiencing pain. And in fact, radiographic findings very poorly correlate with the patient experience. If we think about using imaging as a potential biomarker, which of course is largely our gold standard in musculoskeletal conditions, what we know is that imaging of the joint, of the spine, does not give us the entire picture. And in fact, when we look at data from low back pain, what we see is that advanced imaging for patients without any indication of serious underlying conditions or red flag conditions such as progressive neurologic weakness or fever, doesn't actually improve clinical outcomes. What it does is increase the number of interventions that we do for these patients, but it doesn't impact their functional outcomes. So this tells us we really need to rethink how we are approaching this patient population. So how can we take the information that we know from the basic science side and bring it to the bedside in ways that we can better inform what's happening at the biological level? Well, this is a summary of data that we accumulated over many years, looking at the effect of both inflammatory and mechanical stress at the most basic level, at the cellular level. And what we were able to demonstrate is that cells from the cartilage of the spine, from the intervertebral disc, respond to inflammatory stress with an increase in catabolic molecules and a decrease in anabolic molecules. But what we also found was that different levels of mechanical loading could also impact those mechanistic cascades. And in fact, there were both beneficial levels of mechanical loading that could dampen the inflammatory response and enhance the anabolic response, but there are also traumatic levels of mechanical loading that could decrease the amount of matrix rebuilding and enhance the inflammatory response. But what we also identified was that cells from patients with degenerative changes had a very different response and had a more accentuated response to inflammation, a more accentuated and detrimental response to traumatic levels of mechanical loading, and a less beneficial response to lower levels of mechanical loading. And so at the most basic level, what this told us was that mechanical loading can be both reparative and detrimental, but it depends on the stage of degeneration in that individual biology. So what this suggests is that exercise, or the way we apply mechanical loading in the clinical space, has potential regenerative capacity, but it has to be titrated correctly to that individual biology. So how can we start to get a handle on that? This is data where we looked at a functional spine unit, so the entire tissue now, bone, disc, bone, applied different levels of mechanical loading, and looked at the same type of thing, what happened to the inflammatory and anabolic cascade. And what we demonstrated, very similar to what we saw in cell culture, was that these tissues had both detrimental responses and beneficial responses to different levels of mechanical loading. But what we were also able to demonstrate, and is shown in this graph, is that we could also assay the markers, the molecules that were released from the tissue into the surrounding media. And in this case, we looked at markers of matrix turnover, CS846, a marker of proteoglycan turnover, and CTX2, a marker of collagen II turnover. And what we were able to demonstrate was that in response to loading, these biomolecules changed in the periphery. And so that suggested to us that in addition to being able to study this at the basic science level, we may be able to translate this into something that we could pick up peripherally and use as a tool to guide our treatments in humans. Certainly we can't assay people's bone or disc or other musculoskeletal tissues in real time. So what we need is some type of surrogate marker that could be picked up peripherally. So we studied this in an animal model of intervertebral disc degeneration. This model uses an annular puncture to the intervertebral disc, which then goes through a slow cascade of degenerative changes that very much mimic what we see in humans. And what you can see on the left here is the MRI findings in that animal model, as you would expect. A decrease in the disc height, a decrease in the disc signal, all associated with gradual degeneration. But what we also did was we sampled the blood of these animals over time, and we measured it for a number of different biomolecules. One of those molecules was that same marker, CTX2, which is a marker of collagen II breakdown. And what we were able to demonstrate is that not only was there a more significant difference between the injured animals and the uninjured animals in the release of this molecule into the bloodstream, but it changed even earlier, as early as 3 weeks, even earlier than the imaging findings suggested. So this told us that we may have the potential of using blood-based biomarkers to not only provide information about active disease, not just previous injury that has accumulated over time, but that it also may be even more sensitive than the imaging modalities that we typically use clinically. So we decided to take this into the clinical space. Let's look at this in humans. It's one thing to look at this in a clean animal model of degeneration, but let's look at this in the more messy background that is the human experience. And so we examined older adults with axial low back pain, and we biosampled them. We biosampled their biomarkers that might be related to pain and pain-related function, and we looked for those associations. And what we saw was a number of biomolecules did show associations with the patient's experience of pain and their self-reported pain. What we also did was we looked at combinations of those biomarkers. And what I have shown here are the R-squared values for those associations and the starred values of those that are statistically significant. And what we saw was that small panels or combinations of biomarkers started to show us significant associations with the patient's experience. Things like the McGill Affective Score, which is a patient-reported outcome that measures the type of pain patients are having and ask questions about whether their pain is cruel or punishing or the kind of emotional aspects of their pain. We also saw associations with depression. We also saw associations with their overall pain score, scored on a scale of 0 to 10. And these were markers of inflammation, markers of pain, and also markers of matrix turnover. But what we also demonstrated is that different types of biomarkers or different classes of biomarkers associated with different parts of the experience of pain. For example, neuropeptide Y, which is a pain-related neurotransmitter, was associated with the McGill affective component, again, that measure of if your pain is cruel or punishing, the depression scores, as well as scores about fear avoidance or fear of movement. So very much associated with the emotional component or the experience that patients had related to their pain. We also saw that RANTES, which is an inflammatory biomarker, seemed to associate with their pain and pain-related function, which was very interesting. So their pain on a scale of 0 to 10, their gait speed, and their performance on a short performance physical battery seemed to associate it with this inflammatory molecule. So again, while these are relatively modest changes in terms of the overall explanatory power of these biomarkers to their pain and pain-related function, I'll compare that to our gold standard, which is MRI. So this is that same set of patients where we compared their MRI to their pain score. And this was either whether we looked at this based on a contour segmentation computer program that measured the overall disc area, disc height, and disc signal, or we looked at it based on independent radiologist readings. We saw absolutely no association with the MRI in this patient population and their pain score. We did the same thing with pain-related function, and again, found no association between the findings on the MRI and the patient's experience of pain. So while the biomarkers were relatively modest associations, they were significantly better than the gold standard MRI. And so this gave us confidence that there might be some opportunity here. Certainly, this could be perceived as a phishing expedition. If you look at enough biomarkers, you're bound to find something that shows some association. But what we need to then do is go and reverse engineer this and bring those findings or those potential signals that we see in the patient population, bring them back to the bench and try to unravel the mechanism. And so I'll give you an example on how we have started to look at that to understand, are these biomarkers that we're seeing signals for really relevant to the overall pathophysiology, and just as importantly, can they be used to guide future treatment? I mentioned neuropeptide Y as an interesting potential biomarker. This is a neuropeptide, a neurotransmitter peptide that is involved both in blocking pain signals, but also in improving resiliency during times of stress. This is really important for musculoskeletal conditions because, of course, the pain is not the only thing we need to treat. We need to treat the patient's function. And so how they are coping with their pain and how they are dealing with any functional loss is just as important as treating the nociceptive pain signal itself. So we looked at this to see if we could look at some of these biomarkers as guiding treatment. This is a study that we did looking at peripheral biomarkers in response to an epidural steroid injection for low back pain. We examined a cohort of patients who were undergoing an injection as part of their routine medical care for their low back pain. So 48 subjects who were undergoing a lumbar epidural steroid injection. We purposely did not choose or influence the choice of patients that were undergoing the injection because, frankly, we wanted to see responders and non-responders in order to see if the biomarker was helpful as a predictive tool. We defined responders as an improvement in 50% or more in their numeric pain rating scale, and then we also examined that across other functional measures. And in fact, the patients that reported a 50% improvement in their pain score in this cohort also demonstrated significant improvement in their disability-related scores. So we were confident with this cutoff to define responders versus non-responders. And what we saw in that biomarker, neuropeptide Y, that I mentioned, was a significant difference between the circulating levels of neuropeptide Y between responders and non-responders. And this was very suggestive that this might be important in helping us predict that. Just as importantly, what we also saw was that there was a difference in responders and non-responders based on their genotype. So looking at a single nucleotide polymorphism of the promoter region of neuropeptide Y, which influences the overall expression of neuropeptide Y and thereby the circulating levels, we also saw a difference between the responder-non-responder profile based on genotype. And of course, this is exciting because it can be a much more stable biomarker than is a circulating protein biomarker, but it also is easier to obtain. So you can obtain it with a simple saliva sample as opposed to needing to isolate blood from the patient. What we also saw was that baseline neuropeptide levels were also associated with the change in their disability score and the change in their pain in response to treatment. And so again, demonstrating some very helpful associations that far exceeded any changes that we could glean based on imaging findings. In a separate study where we looked at individuals with spinal stenosis and looked at patients and their response to exercise, we also saw a signal in differentiating the responder-non-responder analysis based on genotype. So in this study, we looked at that same promoter region single nucleotide polymorphism, and based on the genotype, saw differences in the responder-non-responder analysis. What was very interesting, however, is when we looked at this based on the type of care they received, usual medical care versus group exercise or physical therapy, what we saw was that individuals in the usual medical care group really didn't show that genotypic responder-non-responder analysis. It was pretty equal across the board. But what we did see is that in patients who are responders or non-responders to an exercise regimen, either group exercise or physical therapy, did differentiate based on their genotype. So again, this is very important in understanding the role of neuropeptide Y as it might relate to predicting different types of treatment. The first example I showed you was an epidural steroid injection, so addressing the response to a treatment aimed at inflammation. And the second one shows you the response of NPY as it relates to a mechanical type of treatment like exercise. So we saw associations to both. So then that asks the question, can we take that back to the laboratory now and start to tease out what is going on? What is the mechanism of that? Well, what we saw was using that same loading system where we had a bone disc bone in the laboratory and applied different levels of mechanical loading. What we saw that was surprising and somewhat unexpected is that there were not only was NPY released into the surrounding media. The reason that's surprising is because I remind you that these tissues were completely stripped of both central and peripheral nervous system. These are really just the musculoskeletal tissues themselves. But we saw appreciable production of neuropeptide Y. But what we also saw that was important is a difference in the amount of NPY that was released from these tissues depending on the type of loading that they were experiencing. So comparing two different modes of mechanical loading, a large flexion extension versus a large flexion extension with an axial torsion, a very physiologic type motion, what we saw was a difference in the amount of NPY that was released. Taking it one step backwards and going into the cellular level and asking the question, are the cartilage cells of the intervertebral disc of the spine actually responding and producing this neuropeptide Y molecule? And again, the answer was a surprising yes. We saw that there was not only appreciable expression of NPY from the annulus fibrosus cells themselves, but that expression was influenced by both inflammatory stress and mechanical stress. So again, what we saw in the clinic, where there was a responsiveness of this circulating biomolecule in response to different inflammatory and mechanical loading treatments, we were able to see that same effect in the cell culture model. What we were also able to demonstrate was that the annulus fibrosus also expressed receptors for neuropeptide Y. So this gives an example of how we could take that signal that might have initially been seen as a phishing expedition, bring it back to the laboratory and start to tease out some of the mechanisms that then will help guide us in terms of next steps, figuring out how we can now address this pathophysiologic condition, not just use the biomarker as a diagnostic, but actually use it to guide therapy in a targeted and rational way. And that's very much what we intend to do. I'll give you an example from another musculoskeletal condition. This is work in osteoarthritis that was performed by a very talented clinician scientist who trained with us and is now at Northwestern, who was interested in biomarkers as it related to knee osteoarthritis. And in his study, what he looked at is patients who underwent 45 minutes of continuous walking versus, and compared those to patients who in a controlled laboratory setting underwent the same duration, total duration of exercise, 45 minutes, but split up by rest breaks. So 15-minute bouts of exercise separated by 1 hour rest breaks. And he measured, again, circulating biomarkers in these patients to compare the difference between continuous walking from interval-based walking. And what he was able to demonstrate, importantly, was that a biomarker, cartilage oligomeric matrix protein, which is important in the turnover of articular cartilage in osteoarthritis, was increased in the continuous walking group, as was their pain level. So when the patient's pain level in that continuous walking group started to increase, so did their circulating levels of this COMP molecule that is indicative of extracellular matrix turnover. So this is a very important, again, both helpful in terms of thinking about how we mechanistically might be able to titrate exercise-based regimens to limit harmful effects that might be seen from mechanical loading. So again, both helpful from a predictive perspective, but also potentially mechanistically from how we would treat these subjects. Similarly, Fabio Ambrosio, who has been studying a longevity protein called Closo, has discovered that really this protein is much more than a potential signaling biomarker. This has been looked at a lot in longevity research as a molecule that is very strongly associated with aging phenotypes. But clearly it's more than just a biomarker. And in her research, what she's been able to demonstrate is that the levels of Closo in the tissue in response to injury go up in a young animal in response to injury, but that same beneficial effect is lost with aging. And in fact, in some very elegant mechanistic studies that she recently published, was able to demonstrate that the inhibition of Closo disrupted muscle progenitor cell lineage progression. It also impaired myofibro regeneration and impaired the bioenergetics of these molecules. So again, teasing out from a potential biomarker, teasing out mechanisms in a way that will allow us to start to target our therapies much more significantly. So I'll end by just talking about how I think we can start to bring this together. While it is unlikely that there is any single biomarker that's going to lead to a large change in clinical treatment, the way I think we should be approaching this is to really characterize the entire condition, the entire syndrome, and bring all of these aspects, measurable aspects, together in a way that can give us stronger predictive tools. So as an example, going back to that same example I showed you about predicting patients with back pain's response to an epidural steroid injection, we looked at the phenotype of these patients as it relates to their clinical data. So information that was pulled out of the electronic medical record and would allow us to characterize patients as either responders or non-responders based on just their clinical profile. And that's what's shown here in this graph, blue peaks being responders and red peaks being non-responders. So each of these peaks represents a different phenotype or grouping of characteristics gleaned from the clinical profile that helps us understand whether the patients are going to respond or not. And while it gives us some predictive capacity, what I think you can also appreciate is there a fair amount of overlap in some of those peaks, where the blue and the red peaks overlap in areas where we have relative uncertainty or inability to predict. If we look at that patient population and again look at their genetic profile only, again what we see is you can get some separation where you can find responder profiles and non-responder profiles, but again a fair amount of overlap. But now in just this small cohort, if we start to combine those data and the genetic profile, we see the best separation of these peaks or phenotypes in ways that give us the best predictive capacity. And that's exactly where I think we need to go in order to move the field forward for musculoskeletal conditions. And this is precisely the topic of our recently funded Mechanistic Research Center. This is part of the Backpack Consortium funded through the NIH HEAL initiative. And I think that this is going to be a wonderful opportunity for us to do just that, deep phenotype these patients in many different aspects in ways that allow us to combine all of those unique characteristics of the patient while maintaining the interconnectiveness of each of those metrics within an individual that gives us their true phenotype or their true profile that will allow us to better tailor an individual therapy to that person's individual phenotype. The way we're going to approach this at our Mechanistic Research Center here in Pittsburgh is we're going to do deep phenotyping of 1,000 subjects with low back pain biologically, where we're going to biosample them. We're going to collect saliva, serum, spinal tissues. We're going to look at genomics, proteomics, transcriptomics, just oodles of data, and mine that data in a targeted way to help us develop phenotypes. We're also going to phenotype these patients behaviorally. So we're going to get patient-reported outcomes. We're going to get information about their physical activity during the day, their sleep, their smoking, alcohol, substance use, their psychological profile, any depression, anxiety, and the important contribution that has in back pain, and put that all together. And finally, we're going to look at the patients and phenotype them biomechanically. We're going to look at very targeted kinematic profiles using biplanar radiography. We're going to use wearable motion sensors to be able to measure them in the laboratory and also in the field. And then we're going to put that together in performance-based measures of function where we can start to, again, characterize these patients biomechanically. By putting all of these different profiles together for each individual, we hope to be able to identify unique phenotypes that will allow us then in adaptive clinical trials to target our treatments much more specifically to the individual and allow this to finally move outcomes in the direction that we need to see for this important patient population. We have a dream team here in Pittsburgh, a very interdisciplinary team made up of basic scientists, engineers, clinicians, biostatisticians, psychologists, therapists, all working around this problem together and all bringing a very different perspective in ways that I think is going to be both unique and high yield as we tackle this important problem. So with that, I'll just give you a glimpse into the future of what's going on here in Pittsburgh. We are currently in the process of building our new Vision and Rehabilitation Tower, where we're literally going to bring bench to bedside. This new tower is going to focus on innovations and have clinical space immediately adjacent to research space, where the clinicians and researchers are literally bouncing off one another, exchanging ideas regularly. And again, we think that this will contribute to that translational approach of bench to bedside and bedside to bench research in exchanging all of those important questions. So I thank you very much for the opportunity to share some perspectives with you today. And I very much welcome your thoughts and questions during our discussion period. Thank you.